Hi, and thanks for watching Cardiovascular Physiology Part 2 of 3. This lecture will be divided into two different segments in order to keep the file size down. So the first segment will include slides 1 through 78, which we can see here the order of the topics that we'll cover, and slides 1 through 78 will be covered in the first segment, and that means in the first segment, we'll get through number 4, Introduction to the Lymphatic System. So the second segment, we'll pick up with number 5, Venous Return, and the second segment will finish part 2 of 3 of cardiovascular physiology. So part 2 focuses on blood vessels. Just to give you a sense of how extensive blood vessels are in your body, you have approximately 100,000 kilometers worth of blood vessels. And in part 1, we examined an image of a cadaver in which the blood vessels were injected with a latex type material. Perhaps you remember that. It's a head and a neck. And all the other tissues were digested away, and that person was still recognizable as an individual. So we don't typically see blood vessels by looking at the surface of a person, but blood vessels are ubiquitous and dense throughout the body of a person. So when we talk about blood vessels, we're talking about tubes, pipes, through which blood travels, and there are 5 major types of blood vessels. And these 5 major types of blood vessels are distinguished by largely size, the direction of blood flow, and other anatomical features that we'll take a look at in a moment. So you recall that the heart pumps blood, and when the heart ejects blood, it ejects blood into arteries. Arteries are the largest blood vessels in the body. They have very thick walls. They carry blood under high pressure. And we'll talk about both of those in a moment. Arteries branch to form arterioles. So generally arterioles are smaller in diameter than arteries, and arterioles deliver blood into capillaries. Capillaries are the smallest diameter blood vessel, and capillaries are the site of exchange. Only the walls of capillaries are leaky, meaning as blood flows through capillaries, materials can enter the blood and materials can leave the blood. Of all the other blood vessels shown here, only capillary walls are leaky. The others are not. Capillaries, the smallest blood vessels, merge together to form venules. Venules are larger in diameter than capillaries. Venules merge to form veins. Veins are larger in diameter than capillaries, and the 2 largest veins in the body deliver blood directly into the right atrium, the superior vena cava and the inferior vena cava. As we move away from the heart, the blood vessels branch. They become more numerous and smaller in diameter. As we move blood back toward the heart, the blood vessels become less numerous and larger in diameter. Here we compare the anatomy of the wall of an artery with the anatomy of the wall of a vein. And without knowing anything about the structure or function of these two objects, you can clearly see differences. Functionally, arteries carry blood under high pressure and veins carry blood under low pressure. Also, arteries are able to change their diameter to a much greater degree than veins can. And those two functional differences are reflected in their anatomy. If you recall, form follows function. So let's start with the amount of smooth muscle. If we look at the amount of smooth muscle in the artery and compare it to the amount of smooth muscle in the vein, we see that the artery has a much thicker layer. This allows the artery to vasodilate, or to get wider, and to vasoconstrict to a much greater degree than the vein. And changing the diameter of a blood vessel is a way of controlling the amount of blood flow through that vessel. So arteries and arterioles, not shown here, but arterioles also have a fair amount of smooth muscle in their walls relative to veins. The second difference I'd like you to focus on is the presence of elastic tissue in the wall of the artery and the relative absence of it in the wall of the vein. Because arteries carry blood under high pressure, their walls need to be stretchy. They need to be extensible and elastic, meaning they can bulge under a surge of pressure and then resume their original shape. And then the third difference I'd like to point out is focusing on the vein. In the vein, specifically the endothelium, that lining of blood vessels and the entire cardiovascular system, the endothelium in veins includes one-way valves. So in this image, the blood would be flowing from the bottom of the vein to the top of the vein, as the vein is represented here. And that valve will allow blood to flow from bottom to top. But should the pressure gradients permit blood to flow downward from top to bottom, the cups or the cusps of that valve would catch the falling blood and would shut. And so this one-way valve prevents the backflow of blood, and veins require these one-way valves because they do conduct blood under very low pressure circumstances, whereas arteries don't need valves because there's always a high pressure and a pressure gradient that forces the blood to move in one direction as it passes through an artery. This graph shows the pressure in the vessels as blood flows through the cardiovascular system. On the vertical axis, we see pressure expressed in millimeters of mercury. On the horizontal axis, we see various blood vessels leaving the heart and then returning to the heart. So blood leaves the heart in the aorta. It travels through arteries, through arterioles, through capillaries, and it makes its way back to the heart through venules, veins, and finally into the heart through the superior and inferior vena cava. The lines on the graph, there are two of them. Let's start by looking at the red line. When we're close to the heart, the aorta, the arteries, and even in the arterioles, we see a jagged spiking line, a line that has steep peaks and deep troughs. So the jagged nature of this line represents the dramatic changes in pressure that occur with contraction and relaxation of the ventricles of the heart. So at the peaks occur because the ventricles contract, and peak pressure that we measure in a blood vessel, particularly close to the heart, is called systolic blood pressure. The troughs are caused by relaxation of the ventricles. The ventricles relax, they're finished ejecting blood, they're relaxed, and they're able to receive blood once again. So the lowest pressure that we observe in the vessels close to the heart is called the diastolic pressure, where diastole is relaxation. The purple line, the dotted line, represents the average of systolic and diastolic. So we can focus on that purple line and then the continuation of that line through the capillaries, the venules, and the veins. So we can see that there's this continuous pressure gradient throughout the cardiovascular system. As blood travels away from the heart, from the aorta, through arterioles, through the capillaries, into veins, it follows a pressure gradient. Blood is always flowing from an area of high pressure to an area of low pressure. And the pressure in the two largest veins in the body, the superior and inferior vena cava, is almost zero. So this promotes what's known as venous return, blood flow back to the heart, which we'll talk about in a little bit. Capillary exchange is an important topic in the cardiovascular system. If you recall, all cells in the human body need to exchange materials with the environment. And the exchange of materials between blood and body cells occurs across the capillary wall. We will spend quite a bit of time talking about capillary exchange. Let's start by examining the structure of a typical capillary bed. So up top, we have an arteriole. An arteriole can be thought of as a small artery. It includes smooth muscle in its wall, and it gives rise to a capillary bed. In the middle portion of the diagram is a capillary bed. It's a branch structure. There are numerous capillaries there, and at the bottom portion of the image is a venule. So blood flows through the arteriole, through the capillary bed, and into the venule. And from the venule, it makes its way back toward the heart. An important part of a capillary bed is that central vessel that extends from the arteriole to the venule, and that's called a metarteriole. The metarteriole has several capillaries branching off of it. So a capillary bed can vary in size. It can be as small as 10 capillaries or as large as 100 capillaries, roughly, that have all branched off that central metarteriole. And there's the pathway of blood diagrammed from an artery to an arteriole through a metarteriole. From that metarteriole, blood flows through individual capillaries and ultimately into a venule. Capillaries are more numerous than the other types of vessels in the body because that arteriole and that metarteriole branch into capillaries, and this branching serves two important purposes. Number one, it decreases the rate at which blood flows through the vessel. So blood flow through a capillary is much slower than through any other type of vessel. And I'll tell you more about that in a moment. The other important reason for capillary branching is that capillaries have far greater surface area than any other type of vessel in the body. In this diagram, we can examine both velocity and cross-sectional area in the variety of vessels shown along the horizontal axis. And it's that same horizontal axis that we've seen previously. On the left, we start close to the heart with the aorta. We move through arterioles, through capillaries, through veins, and back toward the heart, back into the heart through the vena cava. There are two lines on this graph. The green line represents cross-sectional area, and we can think about that as surface area. And the black line represents velocity, so the speed with which the blood flows through a particular type of vessel. So let's start with velocity. How quickly is the blood flowing through a vessel? And we'll begin close to the heart on the left side of the diagram, the black line in the aorta. And again, we see those steep peaks and deep troughs as blood speeds up and slows down, speeds up and slows down, speeds up, slows down, speeds up, slows down. And those surges of velocity correspond with the cardiac cycle. So with every ventricular systole, contraction, blood's ejected from the left ventricle into the aorta, and there is a surge in velocity. And then the heart relaxes, ventricular diastole, and blood slows down. And as that blood moves away from the heart, eventually those surges fade away, and we get that nice smooth line that we see through the capillaries. And as that blood flows through the capillaries, we can see that the velocity undergoes a steep drop off. So blood was flowing relatively fast through arteries and arterioles, and then the rate of blood flow, the velocity of flow, drops down as we move through the capillaries. And then as that blood continues its way through the venules, veins, and into the vena cava, the velocity picks up again. So blood flows most slowly through capillaries, and that's important. Remember that. I'll talk about that next. Looking to the green line represents cross-sectional area, and we can think about that as a reflection of the amount of surface area offered by each type of vessel in the body. And so the large vessels near the heart, the aorta, the arteries, and the arterioles, have a much smaller amount of cross-sectional area relative to the capillaries. As those arterioles give rise to capillary beds and branching occurs, we see this surge in surface area available. And that surface area is important because exchange occurs across the surface. So the more surface area that's available, the more opportunity there is for exchange. And as that blood continues through the capillaries into the venules, veins, and back to the heart, the cross-sectional area is dramatically reduced. So what is the significance of those two lines in the previous graph? It's important that blood flows through capillaries slowly. The more time that blood passes through a capillary, the more time there is for exchange. Another point of significance is that capillary beds have a high surface area. All exchange occurs across a surface. The more surface area, the more opportunity there is for exchange. To illustrate that point, we can look at these two, and I'll put quotes around that, capillary beds. Which one is better suited for exchange? So if we look at the top panel, that capillary bed consists of just a single capillary. And the bottom panel, there are numerous capillaries there. So there's far more surface area available in the bottom panel relative to the top panel. So in the bottom panel, there's a much greater opportunity for exchange. So the tissue cells in that bottom panel are going to be able to receive more oxygen, more nutrients that make their way out of the blood. And in that bottom panel, those capillaries are also able to take up or receive carbon dioxide and other waste products and carry them away from the tissue. We're also talking about the velocity of blood flow. Why does the velocity of blood flow slow down as it passes through a capillary bed? So if we look at the image, at the top of the image, we see an arteriole. And as blood flows through that arteriole, it's moving fairly quickly. And then as blood passes into that medarteriole and then makes its way through the capillaries, its rate of flow, its velocity slows way down. And then as it makes its way into the venule at the bottom of the screen, it starts to speed up. It flows faster. So how come? Well, it turns out that there is friction between the liquid that's flowing through a tube and the wall of that tube. And that friction is responsible for slowing the flow of blood down. And the degree of friction is related to the diameter of the tube. So if we look at the diameters of the tubes shown here, we have arterioles, we have capillaries, we have venules. Arterioles and venules have a larger diameter than capillaries. There is less what's known as vascular resistance in an arteriole in a larger diameter vessel, less vascular resistance in a venule, also a larger diameter vessel, and there's greater vascular resistance in a capillary, which is a smaller diameter vessel. So resistance is an impeding force to flow caused by the friction that occurs between blood inside the tube and the wall of the tube. Here we're talking about specifically vascular resistance. That's the friction between blood and the wall of the blood vessel. And in the respiratory system, we'll talk about a very similar concept, but it's called airway resistance. And that's friction between the moving gas and the airways, the wall of the bronchi, the bronchioles, etc. So there's a relationship between the diameter of a tube and the degree of resistance or the degree of friction. In this diagram, we can look at the velocity of blood flow through different points in the interior of a vessel. So the length of the arrow represents the velocity of blood flow at that point inside the vessel. So in the very center of the vessel, we see the largest arrow, and blood that's flowing through the center of a tube or a pipe, that blood is free from friction. It's not going to be slowed down by friction against the wall of the tube, and that's known as laminar flow. But as we move away from the center and we approach the edges of the tube, notice that the arrows get shorter and shorter and shorter. The closer that blood is to the edge of that tube, the more it will be slowed down by friction. Here we can compare two vessels with two different diameters and think about how much friction is influencing the rate at which blood flows through each vessel. So up top we have a larger diameter vessel, and again we see a similar setup with arrows of different lengths in different locations inside that vessel. And up top, notice that we see more long, thick arrows near the center of that vessel that are more or less free from the forces of friction. Then down below, we don't see very many arrows that are free from the forces of friction. So up top, in that larger vessel, we have less vascular resistance and a faster flow rate. And down below, in that smaller diameter vessel, we see more vascular resistance and therefore a slower velocity of flow. And that brings us to capillary exchange. So capillary exchange refers to the movement of substances across the wall of a capillary. And during capillary exchange, substances move between blood, that's of course inside the capillary, and interstitial fluid. That's of course outside the capillary. And you remember blood and interstitial fluid are two important body fluids for the cardiovascular system. And this diagram shows the direction in which typical substances move. So as that blood flows from the arterial end of the capillary on the left toward the venual end of the capillary on the right, capillary exchange occurs. Substances that typically leave the blood and enter interstitial fluid are indicated by the red arrows, so water, nutrients like amino acids and glucose, and oxygen. And then substances that typically move in the opposite direction from interstitial fluid into the blood include water, carbon dioxide, and other waste molecules. In order to understand capillary exchange, we need to start with an understanding of the structure of a capillary wall. There are several different types of capillaries in the body. What they all have in common is their walls are leaky. Capillaries are the site of exchange, so there are literally holes in the walls of capillaries. Where they differ is just how leaky are they. So we're going to talk about continuous capillaries. Continuous capillaries have a moderate level of leakiness, and they are the type of capillary that occur in your generic systemic tissue. So continuous capillaries serve muscle tissue and fat tissue and connective tissues all over your body. The capillary wall is, of course, very thin. That makes it easy for substances to travel across it. The capillary wall consists of a single layer of endothelium. Endothelium is the tissue that lines your entire cardiovascular system, the walls of all of your blood vessels, and the chambers of your heart. In the capillary wall, that endothelium is surrounded by a basement membrane. Endothelium is an epithelial tissue. If you recall, perhaps you've taken anatomy, epithelial tissues serve as coverings and linings. So here we're seeing a clear example of it being a lining. And all epithelial tissues are associated with a non-cellular basement membrane. And that basement membrane is shown here in grayish blue. So it's a very thin wall. It's a single layer of simple squamous epithelial cells, these very flat cells, surrounded by basement membrane. These endothelial cells have spaces in between them, and those spaces are called intercellular clefts, and they are literally holes in the walls of capillaries. Here we see a different representation of a capillary wall. Here I see 4 different endothelial cells represented in pink. None of them are touching each other. In the upper left side of this panel, we see an intercellular cleft labeled as a water-filled pore. And the rest of this diagram indicates process, how substances are able to cross the wall. So in this diagram, we see both form and function, both anatomy and physiology. And in this diagram, we can start to see some of the factors that influence the physiology here, how substances move across the capillary wall. We see that size is important. We see that solubility is important. So in the upper right panel, in the upper right corner, we see in blue, it says plasma proteins generally cannot cross the capillary wall. This is due to their size. So proteins are relatively large molecules, and anything that's the size of a protein or larger cannot fit through what's shown here as a water-filled pore or an intercellular cleft. They're too big. And then solubility is also represented here on the right side of the diagram. We see that lipid-soluble substances can cross the capillary wall by one means, and water-soluble substances, small water-soluble substances, can cross through that intercellular cleft. So there are other factors that drive the movement of substances across the capillary wall, but here we're being introduced to size and solubility. Other factors include gradients, pressure gradients, and concentration gradients. So capillary exchange occurs by three different mechanisms, and they all co-occur. As blood moves through a capillary, substances are entering and leaving the blood by all three of these processes simultaneously: diffusion, transcytosis, and bulk flow. So you're very familiar with diffusion. We studied it, I feel like it's day 2 at the very beginning of the semester. Diffusion is the movement of a substance along its concentration gradient from an area of high concentration to an area of low concentration. Here we see the movement of materials between blood, so that's inside the systemic capillary at the top, and a tissue cell. So down below we see a muscle cell, and not really well represented here, but between the blood vessel and the muscle cell we have interstitial fluid. So during capillary exchange, I'll remind you that substances are moving between blood and interstitial fluid. The grayish-blue arrows represent diffusion of substances along their concentration gradients. So let's just go from left to right on this image, starting with carbon dioxide. Carbon dioxide is a waste product. Cells produce carbon dioxide during cellular respiration. As cells catabolize fuel molecules like glucose in order to extract energy and make ATP, the resulting waste product includes carbon dioxide. Got to do something with those leftover carbon atoms. It's carbon dioxide. So cells are constantly generating carbon dioxide, and that carbon dioxide is small, it's lipid soluble, it easily diffuses across the plasma membrane of the muscle cell into interstitial fluid. It easily diffuses across the capillary wall and into blood along its concentration gradient. Moving to the right, we see oxygen. Oxygen is plentiful in blood that enters a capillary, and as shown by the grayish-blue arrow, there's a concentration gradient between blood that's inside the capillary and interstitial fluid, and furthermore, there's an even lower concentration of oxygen inside that muscle cell. Remember, inside cells, oxygen is used to produce ATP. And so there's a relatively low concentration of oxygen inside the cell. There's a relatively high concentration of oxygen inside the blood capillary. And so by diffusion, oxygen moves along its concentration gradient out of the blood through interstitial fluid and into the muscle cell. And then continuing to move to the right, we see glucose. There's glucose in the blood. There's a relatively high concentration of glucose in the blood. If you recall, inside of our cells, glucose is used. It's used and it could be used up as a fuel source for making ATP. So there's a relatively low concentration of glucose inside that cell. So glucose is going to diffuse along its concentration gradient from inside the blood across the capillary wall into the interstitial fluid, from the interstitial fluid across the plasma membrane of that muscle cell and into the cell. Here are two new vocabulary words. We use the terms unloading and loading to generally describe capillary exchange. When substances leave the blood and enter interstitial fluid, we call that unloading. So as blood flows through a capillary, generally speaking, oxygen and glucose are unloaded. They leave the blood and they enter interstitial fluid. And then the opposite of unloading is loading. Loading means the substance moves from interstitial fluid into the blood. So carbon dioxide and other waste substances are loaded during capillary exchange. The solubility of a particular substance determines the exact route it will take across the capillary wall. Water-soluble substances cannot easily pass through cells. They can't easily cross a phospholipid bilayer, a plasma membrane, and so water-soluble substances pass between cells through those intercellular clefts. And examples of water-soluble substances that move in those intercellular clefts are glucose and amino acids. Lipid-soluble substances, if they're small enough, they can easily cross a phospholipid bilayer. They can easily cross through a plasma membrane. And so small lipid-soluble substances can move directly through those endothelial cells. They don't need to rely on passage through an intercellular cleft. They can go right through the wall of the capillary, meaning right through those cells. And so small lipid-soluble substances that are exchanged right across those endothelial cells include oxygen, carbon dioxide, and steroid hormones that are lipid-soluble. Okay, so that's diffusion. Of these three, diffusion plays the biggest role in capillary exchange, but we also have transcytosis and bulk flow. So let's think of this as just a single endothelial cell, just one cell that's making up part of the wall of a capillary. And on the left side of this image is the blood. On the right side of the image is interstitial fluid. And now we're thinking about how a particular protein signaling molecule is going to cross the capillary wall. Remember, proteins are too large to pass through intercellular clefts. They're stuck in the blood, except some proteins need to routinely cross the capillary wall, these protein signaling molecules. Like insulin. Insulin is a protein hormone. It's secreted by the pancreas. It crosses the capillary wall to get into the blood, and it needs to cross the capillary wall to get out of the blood. And many different types of cells in your body have receptors for insulin, are able to respond to insulin. So let's say those blue dots in the blood on the left side of that diagram are protein signaling molecules, specifically insulin. How do they cross the capillary wall? Well, this endothelial cell transports those insulin molecules across it, transcytosis. So on the blood side of that cell surface, endocytosis occurs. Insulin molecules enter the cell as a vesicle forms around it. That's endocytosis. And then that vesicle travels across the cell, and on the opposite cell surface, the interstitial fluid facing cell surface, exocytosis occurs. And so those insulin molecules, those blue dots, are now released from the endothelial cell into the interstitial fluid. And not shown here, but there are obviously tissue cells, and that insulin can then go on to bind to receptors on those tissue cells. So this is transcytosis. Going back to this diagram that we looked at earlier, to understand the structure of the capillary wall, I want to point out that the artist included pinocytic vesicles. These are the vesicles we were just talking about in the previous diagram. They're the vesicles that are engaged in endocytosis. Nope, excuse me. They're the vesicles that are engaged in transcytosis. So endocytosis occurs on one cell surface, exocytosis occurs on the opposite cell surface, and in that way, a particular substance is transported across the cell by means of vesicles. So the example signaling molecule that we're using here is insulin. Again, insulin is a protein signaling molecule, and it's too large to get through those intercellular clefts. But insulin needs to get into your blood as that blood passes through the pancreas, and insulin needs to get out of your blood as that blood passes through all of your systemic capillaries. Okay, so our third mechanism of capillary exchange is called bulk flow. And bulk flow is really where we start to rely on pressure gradients, both hydrostatic gradients and osmotic gradients. So bulk flow is the movement of a whole set of ions and solutes and fluid in one direction. So bulk flow is not particular at all. Substances move as a group. I worked in a hardware store for many years during college and graduate school, and many of my physiology analogies come from items that are available in your hardware store. What we're looking at here is called a perforated garden hose, and somebody might buy that in order to water their garden or to water their lawn. You hook it up to the spigot on the outside of your house, you lay it down in your flower bed, you turn on the spigot, and water will exit the hose over the whole length of the hose, right? So the wall of the hose is permeable, much like the wall of a capillary. And the movement of water here from inside the hose to outside the hose, that's driven by a pressure gradient, right? The water pressure inside the hose is greater than the pressure outside the hose. And so water is going to flow from an area of high pressure to an area of low pressure. And yes, this is water, but let's imagine that this is not pure water. Now we've added a bunch of solutes to it. So it's water that includes sugar, it's water that includes amino acids, it's water that includes ions. And all of those things are going to move collectively. Those holes in the wall of the hose are not selective. So under this pressure gradient, water and all of the solutes that can fit through the holes are going to move collectively. And so that's an illustration of bulk flow. An important difference between that perforated garden hose and a capillary wall is that in the capillary, bulk flow is bidirectional. So when substances leave the capillary and enter interstitial fluid, that's called filtration. And then when substances leave interstitial fluid and enter the capillary, become part of blood, that's called absorption. In this image, we're looking at a simplified capillary bed. On the left, we see an arteriole. On the right, we see a venule. And that capillary bed is represented by just a single capillary for the sake of simplicity. And as blood flows through that leaky capillary, both filtration and absorption occurs. During filtration, substances leave the blood and join interstitial fluid. And during absorption, substances leave interstitial fluid, cross the capillary wall, and become part of blood. Two different types of pressure gradients drive bulk flow: hydrostatic pressure and osmotic pressure. Hydrostatic pressure is more easily imagined. It's more tangible. So if we go back to that perforated garden hose, and what drives the movement of water from inside the hose, across the wall of the hose, to outside the hose. That's completely a hydrostatic pressure. There's water pressure. A hydrostatic pressure is the pressure that's exerted by a liquid against the wall of its container. So you turn on the spigot, water pressure goes up, and water shoots out of the hose. Blood also has a hydrostatic pressure. It's a liquid confined to a tube, and so hydrostatic pressure is caused by blood pressure, this constant beating of your heart that gives rise to pressure. Osmotic pressure is a completely different type of pressure. Osmotic pressure is a pressure that's caused when we have some type of solute that's stuck or trapped on one side of a semi-permeable membrane. And so the image here, you've seen similar images before, right? We studied osmosis, osmotic pressure, osmosis, the movement of water. There's a link here. So an osmotic pressure is caused by solutes that are stuck on one side of a semi-permeable membrane. In blood, osmotic pressure is generated by plasma proteins. There are many different types of proteins that are part of plasma, and they stay in the plasma. They're not signaling molecules. They don't cross the capillary wall. But instead, because they're stuck, they give rise to an osmotic pressure. In this diagram, we can see a semi-permeable membrane, that pink dotted line in the middle. Only now we're using that semi-permeable membrane to represent the capillary wall. And down below, we see blood plasma. Blood plasma includes many different solutes. Some of them are proteins. Proteins are represented by the larger dots here. And proteins are too large to pass through those intercellular clefts. Smaller solutes like glucose and amino acids can pass through those intercellular clefts and can freely cross, but the proteins cannot freely cross. And so they give rise to an osmotic pressure because they are solutes trapped on one side of a membrane. And these trapped solutes tend to attract water molecules toward them. And so they play an important role, these plasma proteins play an important role in limiting filtration and promoting absorption. These plasma proteins, because they attract water molecules to them, they slow down the rate at which water leaves the capillary, and they increase the rate at which water enters the capillary. So they slow filtration and they enhance absorption. So let's revisit this image. Notice that as blood flows through that capillary, there's more filtration that happens on the arterial side of that capillary, and more absorption happens on the venule side of that capillary. So these pressures, the hydrostatic and osmotic pressures, they need to necessarily change their balance, change their relative contributions. And that's going to account for more water and solutes leaving initially, and then more water and solutes entering the blood later on. So the entire length of a capillary, the balance of hydrostatic forces and osmotic forces determine whether or not filtration prevails or absorption prevails. And so this diagram comes from your textbook, and let's look at the four forces that determine bulk flow at any particular points along the length of a capillary. Two of the forces are osmotic forces, and two of them are hydrostatic forces. And let me remind you that we're dealing with two different fluids here. We're dealing with blood that occurs inside the capillary, and we're dealing with interstitial fluid that occurs all around outside the capillary. So let's just examine these four forces moving from left to right. Two of them are hydrostatic, two of them are osmotic. So starting with capillary hydrostatic pressure on the left. It's abbreviated capital P sub C. So that's a hydrostatic pressure that occurs inside the capillary. That's blood pressure, right? And the pressure of the liquid inside the vessel is higher than the pressure of the liquid outside the vessel. And so that's going to promote, as indicated by the arrow, capillary hydrostatic pressure is going to promote filtration, bulk flow, the movement of water and solutes out of the capillary. Moving over to the right, our next force is represented by capital P sub IF. This is also a hydrostatic pressure. It's the hydrostatic pressure of interstitial fluid. So this is the pressure that interstitial fluid exerts against the exterior wall of the capillary. And so that pressure promotes the movement of water and solutes out of interstitial fluid into the blood. So that's absorption. Next, moving to the right, we see the osmotic force due to plasma proteins. Osmotic force, sometimes called oncotic force or oncotic pressure, is often represented by the symbol pi. So pi sub C is the osmotic pressure that we observe coming from inside the capillary. There are these proteins that are stuck inside the blood and they're attracting water molecules to them. And so they promote absorption, as indicated by the arrow, the movement of water and solutes into the blood. And then finally, moving one over to the right, we see pi sub IF. So that's the osmotic pressure of interstitial fluid. Are there proteins in interstitial fluid? Not very many. There's a little bit. There are far more proteins in blood. There are very few proteins in interstitial fluid, but they're there. We need to account for them. And so whatever proteins there are in interstitial fluid, they're also exerting an osmotic pressure. They're stuck in interstitial fluid. They can't cross the capillary wall either. And so they tend to attract water molecules. And so the very few proteins that are present in interstitial fluid, they're attracting water molecules toward them. And so they influence bulk flow. Specifically, they promote filtration and they retard or inhibit reabsorption. So of these four forces, two of them promote filtration. That is the movement of water and solutes from the blood into interstitial fluid. And those two filtration promoting forces are capillary hydrostatic pressure and the osmotic force caused by those proteins that are in interstitial fluid. Here are those filtration promoting forces in our diagram. The other two pressures promote absorption, the opposite movement, the movement of water and solutes from interstitial fluid into the blood. And those two filtration promoting forces are interstitial fluid hydrostatic pressure and the osmotic force caused by those plasma proteins in the blood. And here they are on our diagram. So for any point along the length of a capillary, we can add up those four forces and determine net filtration pressure. And net filtration pressure will tell us whether or not filtration's happening at that point or absorption's happening at that point. Net filtration is determined by adding together the two forces that promote filtration, and from that, subtracting the sum of forces that promote absorption. If we obtain a positive value for NFP, by convention, that means that there's a net movement of fluid and solutes into the tissue. So there's a net filtration. If we obtain a negative value for NFP, it means there's a net movement of fluid and solutes into the blood, into the capillary. So there's net absorption. And so we'll do a couple of examples here using two different points along the length of the capillary. We'll calculate NFP somewhere closer to the arterial end of a capillary, and then we'll calculate NFP somewhere closer to the venule end of a capillary. So let's look at the arterial end of this capillary. We see our four forces represented. Moving from right to left, we see P sub C, that's hydrostatic capillary pressure, so that's blood pressure inside the capillary, and that has a value of 35 millimeters of mercury. We see down below, we see the hydrostatic pressure of interstitial fluid, and that's very low. It's so low that in this example it's 0. That's the pressure with which the interstitial fluid pushes against the exterior of the capillary wall. So here that's 0. To the right, we see the osmotic pressure inside the capillary. That's the pressure generated by the proteins that are part of blood, and that's 28 millimeters of mercury. And then our fourth force is the osmotic pressure in interstitial fluid. That's very low. It's not 0 here. There is some protein in interstitial fluid. Here we've assigned it, for this example, we've given it a value of 3 millimeters of mercury. So down below on that arterial end of the capillary, we do the math where we sum together the forces that promote filtration and we subtract the sum of the forces that promote absorption, and we end up with a positive value. It's 10 millimeters of mercury. So because this is a positive value, it tells us that for this point along the length of the capillary, if we add up all of those forces, we have a net filtration pressure that indicates filtration is happening. So more fluid is leaving the capillary than is entering the capillary. Okay, now we can move to a different point on the length of this capillary. On the right side of this diagram, we're looking at a point closer to the venule side. So blood is flowing through this capillary. It's moving from the arterial toward the venule. And we see that the forces are a little different here. We see the same four forces, but they have different values. So P sub C, that's the hydrostatic pressure that blood exerts against the wall. P sub C is now 15 millimeters of mercury. This blood has lost actually a lot of fluid, so the pressure goes down. We went from 35 millimeters of mercury on the arterial end to 15 millimeters of mercury on the venule end. Our second force down below is PIF. That remains unchanged. So for this particular tissue, there's essentially no hydrostatic pressure being exerted by the interstitial fluid against the wall of the capillary. Moving on, we see pi sub C. That's the osmotic force caused by plasma proteins. Well, the concentration of plasma proteins in blood hasn't changed. As we move from the arterial end toward the venule end of a capillary, the concentration of proteins remains the same. Those proteins are stuck in the blood. And so the osmotic pressure in blood stays the same. In this example, it's 28 millimeters of mercury. And that's also true for the osmotic pressure of the proteins that are part of interstitial fluid. So pi IF is 3. Those proteins are stuck in interstitial fluid. They can't go anywhere either. And so if we do the math for the venule end of the capillary, we add together the forces that promote filtration, and from that we subtract the sum of the forces that promote absorption, and we end up with a negative number. Negative 10 millimeters of mercury tells us that at this point along the length of the capillary, and we're near the venule end of the capillary, these forces sum in such a way as they promote absorption. So at the venule end of the capillary, more water and solutes are going to leave interstitial fluid and enter the blood. So now that you have an understanding of the four forces that determine whether or not filtration versus absorption are happening, I need to make an adjustment to this diagram. This diagram is incorrect in a really important way, and it brings us to our next concept related to capillary exchange. So in this diagram, the way it was originally created, notice that on the arterial end of the capillary, net filtration pressure adds up to positive 10, favoring filtration, and on the venule end of the capillary, net filtration pressure adds up to negative 10, favoring absorption. The way this diagram was originally created is such that filtration on the arterial end equals absorption on the venual end. And that's not an adequate reflection of reality. In reality, these forces on the venual end sum to a slightly lower net filtration pressure than on the arterial end. And we can use a diagram taken from just a different physiology textbook to illustrate what I mean. Before we move over to a new diagram taken from a different textbook that uses different terminology, let me make the correction using this diagram that we've spent now a lot of time with. So notice that I've changed the values. Specifically, I've altered the hydrostatic pressure inside the capillary, P sub C, at the venual end of the capillary. By making that a little lower and doing the same math, we see that net filtration pressure sums to not -10, but -9. So this is going to have the overall effect if we now think about the entire length of the capillary and the balance of filtration versus absorption. Across the entire length of the capillary, we're going to have a little bit more filtration than we do absorption because of this difference. A little more fluid is going to leave the capillary than enters the capillary. To illustrate that point again, I've selected a diagram out of a different textbook. This is a textbook also covering capillary exchange and those same 4 forces that determine net filtration pressure, and we use to determine for a particular point along the length of a capillary, is filtration occurring or is absorption occurring? So the terminology here is a little bit different, but I want to take you down to the bottom of the image where here we have those 4 forces at the arterial end, and they sum to +10 millimeters of mercury, which means those forces favor filtration, and that's represented here by this purple arrow showing that more water and solutes are going to leave the capillary by bulk flow. And then let's look at the venous end and the values that are used here in this textbook. The values they use mean net filtration pressure at this end of the capillary sums to -9 millimeters of mercury. And the negative number indicates that absorption is going to occur. But again, the important point here is that over the entire length of the capillary, there's a little bit more filtration than there is absorption. So a little bit more fluid leaves the capillary than enters the capillary. And so we end up with a little more interstitial fluid and a little less plasma. So to summarize bulk flow during capillary exchange, we can use this simplified diagram. Notice those 4 forces are not represented here. Instead, there's a summary of those 4 forces. So at the arterial end of a vessel here, ultimately, blood pressure is higher than osmotic pressure, and that allows filtration to occur. And as that blood travels the length of the blood vessel, the conditions change. As the blood loses water and small solutes, the forces sum differently. At the venous end, the osmotic pressure inside the blood vessel, due to those proteins, the osmotic pressure is higher than blood pressure, and that's going to promote absorption. So here's Starling's law of the capillaries, and it's the same Starling physiology scientist, as was discussed in the Frank Starling law of the heart. So the same Starling. And according to Starling's law of the capillaries, the volume of fluid and solutes absorbed is almost as large as the volume filtered. And so here are some typical values. On average, 85% of the fluid that leaves the capillary is reabsorbed. So that leaves 15% remaining as tissue fluid. Well, what happens to that extra tissue fluid? It will be taken up by lymphatic capillaries. Many people have no idea what the lymphatic system is when they step into a physiology class. Here's our introduction to the lymphatic system. So before we move on to that, think about this. In a given day, in a typical human body, 20 liters of fluid leave the capillaries by bulk flow. 17 of those 20 liters return to the capillaries by bulk flow. And 3 liters, that remaining 3 liters, are going to not stay as part of interstitial fluid, but rather they're going to enter lymphatic capillaries. So here we're looking at a capillary bed. We see tissue cells, and we see that green vessel there. That's a lymphatic capillary. Lymphatic capillaries transport lymph. Lymph is an extracellular fluid. There are no cells in lymph normally. Lymphatic capillaries take up excess interstitial fluid, and they will ultimately return it back into blood plasma. Lymphatic capillaries are blind-ended capillaries. Notice down here, let me change colors. Notice down here, this capillary ends, it terminates. It's not connected to anything. That's what it means by blind-ended. The walls of this lymphatic capillary are especially leaky. It's very easy for water, solutes, and even cellular debris, bacteria, viruses, broken parts of cells to enter this lymphatic capillary. And then as indicated by the arrows, that lymph is going to flow. It moves through these capillaries. It's going to travel through your lymphatic system and ultimately be filtered and then dumped back into blood plasma. So here's an overview of the lymphatic system. The lymphatic system is characteristically represented by artists in green, just like the nervous system is characteristically represented by yellow. And nerves in real life are not yellow. Lymph and lymphatic vessels in real life are not green. Lymph is a pretty colorless fluid. And the smallest lymphatic vessels are lymphatic capillaries, and they are widely distributed throughout our body tissues. And their primary role is to take up excess interstitial fluid and cellular debris and to transport that fluid, now it's called lymph, to transport that lymph through a series of lymph nodes, and these lymph nodes act as filters, to transport that lymph through a series of lymph nodes, and ultimately that lymph is going to be returned into the blood. And I'll show you how. So there are lymphatic capillaries, let's say down here in your gastrocnemius, that take up excess interstitial fluid. Now that fluid's called lymph, and that lymph is going to travel through a series of lymphatic vessels, and these vessels are getting larger and larger. Certain places in your body you have high densities of lymph nodes, like your inguinal lymph nodes here. And as that lymph passes through a lymph node, it gets filtered. Lymph nodes, I'll show you an image in just a moment. Lymph nodes are full of white blood cells that are on the lookout for viruses, bacteria. They're also able to degrade cellular debris. So they really filter and cleanse that lymph. And that lymph travels through lymph node after lymph node being filtered, and it ultimately gets returned into your blood as lymphatic vessels merge with large veins that are in your thoracic cavity. The subclavian veins are where lymph is dumped into blood, so that lymph is returned to blood plasma. Edema is the accumulation of excess interstitial fluid. Maybe you've observed edema on your own body at some point. You've had swollen ankles or swollen feet. Perhaps you've spent a long time on your feet. Perhaps you've been through a pregnancy. Sometimes edema is an indicator of an underlying disorder, so it's an important diagnostic clue. And sometimes edema is not necessarily a reflection of a problem in particular, but edema occurs when there is a change in the balance of those forces that determine bulk flow, filtration, and absorption. Edema is caused when interstitial fluid accumulates. So what's going to lead to that? What's going to cause the buildup of tissue fluid? So tissue fluid will build up when the amount of capillary filtration exceeds by a lot the amount of capillary absorption. So edema can result from either too much filtration or not enough absorption. In either circumstance, we're going to have this accumulation of interstitial fluid. So what might cause excess filtration? What might cause additional fluid and solutes to leave the capillary and join interstitial fluid? Well, we can have an increase in blood pressure. That's the hydrostatic pressure of blood against the capillary wall. And that's the primary driver of filtration. Hydrostatic pressure forces water and solutes out of the capillary and into interstitial fluid by bulk flow. A second factor that can increase filtration is an inflammatory response, which I don't think we've talked about before. So inflammation is a normal response on the part of your immune system. And part of inflammation involves increasing the permeability of the capillary wall. And when we make the capillary wall more permeable, it means that water and solutes can more freely cross that capillary wall. And in some instances, the capillary wall can become permeable enough to allow proteins to cross. And there are far more proteins inside the blood than there are in interstitial fluid. So proteins during the inflammatory response can leave the blood and enter interstitial fluid. That, of course, is going to alter the balance of those four forces that influence bulk flow. Remember, two of them were hydrostatic, two of them were osmotic. So when that capillary wall becomes permeable to proteins, those proteins are on the move and they're no longer solutes trapped on one side of a semi-permeable membrane. And so this is going to allow more water and solutes to leave the capillary and build up in the tissue as interstitial fluid. And then we can also cause edema through inadequate absorption. So what's a factor that will lower the movement of water and solute from interstitial fluid back into the blood? And the most obvious cause of inadequate absorption is having too few plasma proteins. Remember, those proteins that are present in the blood are normally, so we're no longer talking about inflammation here, but under normal circumstances, the capillary wall is not permeable to proteins, and those proteins that are part of blood plasma exert a very important osmotic force. It's the driving force for absorption. It pulls water and solutes back into the capillary. And so if we have too few plasma proteins, then we have insufficient absorption, which means that more fluid remains as interstitial fluid and it can build up over time, causing edema. These are some reasons why a body might have insufficient quantities of protein in their plasma. So plasma proteins are normally synthesized by your liver and added to the blood. So if somebody is suffering a liver disorder, they might have low plasma protein. Proteins are manufactured constantly by most of our cells, and the building blocks of proteins are amino acids. If somebody is malnourished, particularly if they have a protein deficiency, they don't have enough building blocks to make protein, and the person might have too few plasma proteins as a result. And then a third cause of low plasma protein might be due to, you can think of it as leaking proteins out of the body at the kidney. We haven't covered the urinary system yet, but normally as blood is filtered by your kidney, proteins are left in the blood. Normally there are no proteins in urine, or very few proteins in urine. The kidneys are supposed to leave the proteins in your blood. But if there's damage to the kidney or inflammation in the kidney, that function can be compromised and we can start to leak plasma proteins and lose them from the body in urine. And if we lose them at a rate that's faster than the liver can resynthesize them, then we can end up with having too few plasma proteins. So to reduce swelling or to prevent swelling, to prevent edema, particularly post-surgically, one approach is to wear a pressure garment. And I know they look really silly, but the way pressure garments work is they increase interstitial fluid hydrostatic pressure, the pressure that the tissue fluid exerts on the exterior of the capillary wall. Remember, that's one of those four forces, and it promotes absorption. So it causes more fluid to leave interstitial fluid, cross the capillary wall, and join blood. So these pressure garments can reduce or prevent edema by increasing hydrostatic pressure in the tissue. So before we move on from capillary exchange, I want to take a moment to really solidify some of the ideas that we talked about around the lymphatic system. So here's a very quick overview of the lymphatic system with particular reference to its importance in capillary exchange and the movement of fluid between different fluid compartments. So one of the major functions of the lymphatic system is to return excess interstitial fluid to your blood, and it filters that in the process. So we've seen both of these pictures before. Let's start with the panel on the left. We're looking at a capillary bed in a tissue. We see tissue cells, and we also see a lymphatic capillary represented in green. I hope you remember that during capillary exchange, there's an imbalance between filtration and absorption. A little more fluid leaves the capillaries, then enters the capillaries. There's a little more filtration and a little less absorption. So what happens to that excess interstitial fluid? It enters the lymphatic capillary. And once it enters that lymphatic capillary, now we call that fluid lymph. Lymph is a fluid that occurs inside lymphatic vessels. And those lymphatic vessels, moving to the picture on the right, those lymphatic vessels are all over the body, and they channel, they conduct, they transport lymph all back in the direction of your subclavian veins. Your subclavian veins are up here, below your clavicles. That's what subclavian veins mean. And they're veins. So they're transporting blood back toward the heart. And so as this lymph, I did this earlier, but just to recap, as lymph, let's say, enters a lymphatic vessel in your leg, it moves through a series of lymphatic vessels. It moves through a series of lymph nodes where it's filtered, and it will ultimately be returned to blood plasma in your subclavian veins. So here we're looking at a lymph node. A lymph node is full of a variety of different types of white blood cells. And white blood cells, there are several different types. They all have their unique functions, but what they all have in common is they're part of your immune system. So these white blood cells are able to, some of them are able to engulf cellular debris by phagocytosis. Some of them are able to recognize pathogens like bacteria and viruses, and by recognizing these pathogens, they're able to initiate an immune response. So lymph nodes are an important component of your immune system, and they serve to filter lymph. So notice on the panel on the right, notice these arrows here. These arrows indicate the movement of lymph into the lymph node. Inside the lymph node, we see this variety of cell types indicated up here. These are all different types of white blood cells. And then that lymph exits the lymph node here, as well as here, and continues on its way toward the subclavian veins. Here we're looking at where the lymphatic system joins the cardiovascular system. So in green, we have lymph lymphatic vessels represented, and in blue, we see the cardiovascular system, blood veins represented. So recently filtered lymph is dumped back into blood plasma here and dumped into blood plasma here. So imagine you're a water molecule in the body and you spend a good part of your day circulating around the body as part of blood plasma. Whee! You go through the heart, you go through arteries, you go through arterioles, through a capillary bed. You don't get filtered or absorbed this time. You end up in a venule, you end up passing back through the heart. And the next time you circulate through a systemic capillary, you get filtered, but you don't get reabsorbed. So now you, the water molecule, you've crossed the capillary wall, but you haven't crossed it back again. So now you're part of interstitial fluid. And you hang out as interstitial fluid for a little while, a few minutes, or maybe a few hours, or maybe a few days. And at some point, you cross the wall of the lymphatic capillary. And now you, the water molecule, you're part of lymph. And you travel through a series of lymphatic vessels that get larger and larger. You pass through several lymph nodes that filter the lymph. And then as the lymphatic vessels connect with the subclavian veins, you're once again part of blood plasma. So you, the water molecule, circulate through different fluid compartments in the body. And those fluids are named according to the compartment. Blood plasma is inside the cardiovascular system. Interstitial fluid is outside the cardiovascular system and also outside of cells, right? It's tissue fluid. And then lymph is a fluid inside of lymphatic vessels. So this is where we'll stop in the first segment of cardiovascular physiology part 2. Pick up with the second segment and the final segment of cardiovascular physiology part 2 next. Thanks for watching.